second part, which will dedicated to statistical analysis. So there will be two speakers today. Myself, I'm Uthairat Nanakorn. I'm a fish geneticist by training and the editor-in-chief of ANRES. Okay, and then after the break, we will have Associate Professor Skon Kunawitron from Animal Husbandry, Professor University also. Associate editor to take care of all the statistical check. He will give a talk on mistake on statistics, which is very interesting and very useful. Outline of the talk will simply started with introduce ANRES to you. Some of you may publish with us, but anyway, it's better to emphasize once again. Then I will go to the main issues of my talk today, which are 5 main reasons that throw your manuscript in the trash bin. Okay, I suppose that all of you don't like this, but you have to listen to carefully. Okay, and then I will have a short break. After that, Dr. Skon will talk about frequent mistakes in statistical design and analysis. So during my talk, if you have a very serious question you want to ask right away, you can turn on the microphone and shout out, let me know. And then at least after my talk before the break, we will have a short period for the Q&A. Okay, I would like to have a response from you because sometimes, you know, what I'm going to talk is ANRES point of view. Once in a while, you may have different idea, different experience, different point of view. Please share with us. Okay, I will go quite fast on this because some of you may have more agriculture and natural resources already. Our journal has a long history of about 58 years, but with the former name, Kasetsat Journal Natural Science, we have changed the name recently, something like 5 years ago. And then we proudly inform you that we are in Q2 in the Science Marco Journal ranking at the moment. Going to Q2 is not easy. It means among the journals in this field in the world, ANRES is among 50 highest percentage of journal that in terms of quality. Okay, so publish with us is quite tough, but it's worth. The aims and scope is quite broad, so it's everything about agriculture and natural resources. But anyway, please bear in mind that only the research, okay, so let's apply the only research results using scientific approach are accepted for publication. Okay, the agricultural research using social science approaches is not accepted. For example, research that you collect data from questionnaires, from interview, from kind of literature review, except the review article. Okay, it's not accepted. We still have to do the upfront rejection quite frequent with this kind of manuscript. So Once you got accepted, we kindly request you to pay $200 acceptance fee. But fortunately, I don't think it's fortunate because of the COVID. We all of us are struggle with financial, so we give you a discount by half to $100 until December 31st. Okay, so if you want to take this benefit, do submit within this year. You may wonder how long would it take for your manuscript to be accepted. So it's approximately 5 months. It depends on your quality of the manuscript and also your luck. If you are lucky enough, reviewer love to read your manuscript, then you will get the result very fast. Some people may have to wait 3 months in order to have enough number of reviewers, so it would take long. And since we are very kind to our authors, so we allow you to make a second revision, third revision, fourth revision. So it takes very long, but it's our policy now to try to make the process shorter. So we would kind of try not to allow you to revise more than twice. So be careful. At the second revision, you have to revise carefully, otherwise you will get rejected. Okay, so come to the main issues of the talk today. It's about 5 main reasons that throw your manuscript to a trash bin. The first one is very important and unbelievable. It happens quite frequent. Around, I can say roughly about 20% of the paper that got rejected from ANRES was because of this reason. The reason is unjustified or lack of research questions. For example, you just repeat what others have done without clarifying the research gap. Okay, you may read the literature and found that people in the Western countries, they use this technology to kind of culture the fish, and fish people always have examples on fish. So you use exactly the same technology and culture your fish, but you did not clarify why you have to repeat the research once again. Are you going to find something new in here, or you just repeat and then you get the same result? If it happened like the last scenario, means you repeat the technology and then you did not get anything new, then your research is not justified. Okay. The other type of unjustification is about reporting data without scientific question. This happens very often. People may collect samples of fish again, or oyster, okay, in different area, and then report the number of bacteria in the oyster. That is a useful information, but it's not. It doesn't contribute to scientific knowledge. Okay. If you want to generate some knowledge, you should have a clear question. For example, does the number of bacteria relate to water quality or relate to season, relate to whatsoever? You collect those data and then you analyze and show, oh, this is my finding. The number of bacteria would change according to temperature. And again, if this thing has already reported, nothing new, that is also your research is justified, but you don't generate new knowledge, it's also not acceptable. Okay, so be careful in this step before you clarify your work as a research. You must have a specific question, and that question should be scientific. Okay, some people may want to kind of turn the routine work into research. That is quite difficult to publish in international journal, especially Q2 journal. Okay, you must be very keen in order to put some scientific question into your work. Okay. The practice that will help you solve this problem is that you should read relevant literatures as many as possible to identify the research gap. Okay, this is the best way to start doing research. I always use this example, maybe some of you may have heard, but this time in order to make it a little bit more interesting, I use the pictures instead of only the words, you know. So all of you may know this prawn already. This is freshwater prawn. The male is much larger than female. Okay, so farmers want to culture all male prawn, and the male prawn has a chromosome as ZZ. So it is contrast with human system. Okay, we have male human have XY chromosome, but this prawn got CC. So there was a publication from Israeli group that if they remove the androgenic gland of the very young prawn, androgenic gland is the gland that produces male hormone. So it will control the sex of this prawn to have male morphology, and also, it is a true male. Okay, but at the very early stage, if you remove the androgenic gland, then you will have the CC prawn, but it becomes female. Okay, can produce egg, behave totally like female. And the removal of the gland should occur during 30 to 60 days post-hatch. And if you take this sex change female, okay, but still have the ZZ chromosome and mate with the normal male, you will get all male first larvae, okay, because they are all have the ZZ chromosome. So this knowledge is already has been published by Israeli group many years ago. But one of my students came to me and told me that, Professor, I want to work on the production of all maple larvae using this technology. So I told him, no way. It was already published. But the student told me that I have read all the paper, including the one that you mentioned about. But I found that they define the period that is appropriate to remove the androgenic gland. It's very long period, 30 to 60 days post-hatch means 30 days. So there is still a room for him, for me, I mean, when he talked. For me, if I can identify the exact period during this time, make the period a little bit shorter. So the more removal of androgen gland will be more efficient, maybe more precise. Okay, so I told him, go ahead. You already identified the research gap already, so you go ahead. And finally, he spent around 6 months and he got a very interesting result that the better, the more precise period to remove the androgenic gland is something about 45 to 50 or something days post-hatch, which is very narrow, much narrower than this one. So we got published with Q1 journal, high impact factor, and also this research is very useful. So this is a very good example for finding the research gap. Okay, so do this every time before you start doing research. Come to the second reason that made your manuscript unacceptable for publishing. Since we are international journal, so we don't accept the manuscript with the topic that is local, is of local interest. Okay, this topic is kind of, there are two types of flaw in this context. Some is really local, could not fix anymore, so that one will be rejected right away. There is another type that actually it can be international, but because of the poor writing that make this look very local. Okay, so I will talk about the second type further. Okay, for example, this is the manuscript from my former student. She submitted to one journal, and fortunately, I became a reviewer. So her title is DNA barcoding of 5 fish species in Essex River, Thailand. So if you just look roughly, it's like, oh, this is very specific to Thailand. And she even started her introduction like XX River located in Thailand. Oh my God. Who wants to know, international readers just don't care where this river is. But in reality, the technology she used is very interesting. Even in the international community, DNA barcoding is still hot up until now. So why don't she just change the title into DNA barcoding of 5 species? No need to put the word, the name of the river and the country of origin, because these 5 fish species, no matter they are in Thailand, they are in Indonesia, they are in Spain. If this species, if they are this species, the DNA would be the same or almost 100% the same. Okay, so just say DNA coding of 5 fish species. That's enough. Then in the introduction, you should start with something that is of global interest, like problems of morphological identification and how does barcoding solve the problems, why these 5 species, why you choose these 5 species. Most importantly, you have to notify what has been done and what haven't been done. Okay, then go to the objectives of the current study. This is fine. So in general, you should avoid having names of locations in the title and in the first paragraph of your manuscript if possible, except in some research. It's like you study the fish assemblage in Lake Victoria. Sometimes the place is very unique. You have to tell the name of the place, the location. Okay, so there are always some exceptions, but generally, if not necessary, no need to put the name of the place. The second example is the manuscript about the one kind of plant which is endemic to Thailand, found only in Thailand, and this plant contains antibacterial substance. Okay, so young researcher may start the introduction like plant X is endemic to Thailand and it contains antibacterial substance. It looks a little bit very local, you know. Some people may start with Thailand is a hotspot for plant diversity. It's not that attractive, you know. Why don't you start the introduction like raise up the issue that is very that can draw international attention? It's very important. It's very crucial. Like nowadays, problems on antibiotic resistance of pathogens are threatening healthcare system worldwide. That is kind of this topic should be very beneficial, especially for the healthcare. You know. Then go further. Natural antibacterial substances may be a good alternative. Blah, 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 blah. Okay, so the same context, the same manuscript, but you can make it local or make it international. Depends on how the writing style. To write a good introduction. Introduction is very important, you know. For me, since I was trained as a fish geneticist, or maybe I can clarify myself as a professor in aquaculture. I don't know other field. If you for the manuscript on corn, on sorghum, on chicken, whatsoever, I don't know in detail, but I would look at a glance of the introduction, whether you have a research question and is the work justified. So introduction is very important. To write a good introduction, you have to know the definition of this part. Introduction is an explanation of how important your research questions are, including some background of the topics. It should contain a clear and concise literature review to show present status or state of the art, and then focus on why do you have to work on this topic. Okay, this is very important, and it should be well organized, not too long. Every sentence should have important meaning. Okay, convince the reader to believe that your research question is important. The introduction will have general structure like this, but there are exceptions. Some people may have 3 parts only, or I mean 4 parts, but within 3 paragraphs, or some people may have 5 paragraphs, but the section there are 4 main sections like this. You should start your introduction by writing about the importance of the issue or the species in general. Okay, if your species is not so important, it's of local interest, grown only in Thailand or grown by ethnic group up on the mountain, then raise up the issue in order to draw attention from international arena. Okay, but if your species is very important, like marine shrimp, and there are so many problems on that, raise up the species first. Okay, then go to the importance of your topic. And the third paragraph is the most important one. You have to review carefully to tell the reader what has been done, what hasn't been done. Okay, and then go to the objective of your study. I have an example. This is my manuscript. Master student is the first author. Okay, so we report the genetic parameters on of the trait of 2 traits, body shape and deformity, in snakeskin gourmet, in placid snakeskin gourmet. So the fish itself is not important at all in the international platform, you know. Okay, it's important to Thailand, but not that much. So, in order to draw attention, I start the introduction by raising this issue first. Body shape is an important trait for fish production. Okay, and then try to relate this with important species, like, oh, this is also important for the species like night tilapia. Okay, this is a big commodity. Barramundi, this is Asian sea bass. So the flatfish, also very important for water species. And then snakeskin gourmet. See, my unimportant fish is kind of grouped with the important one under this umbrella. Under the body shape is an important trait. Okay, so it's like we can persuade or convince the reader to see, oh, this is important. Okay. So therefore, genetic aspect of this trait have received much attention, blah, 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 blah. And then in the second paragraph, so in my introduction, the first and the second section, I merged into the first paragraph. Okay, start with general importance, which is body shape, and then draw a little bit to our to my topic, which is study on genetic aspect, genetic parameter whatsoever. So two sections were in one part, in one paragraph. And then in the third paragraph, I review intensively about the study of body shape in different kind of fish by telling that what kind of parameter they use to measure the body shape, and then what has been done. Then go to the second trait, which is deformity. So I do the same way. Deformity is another important trait that adversely affects the value of fish production. Okay, and then give them the state of the art. Okay, this in the last paragraph, I mentioned my fish because it is important to tell the reader why you are working on this species, why you choose this species. Okay, if even though this species is not internationally important, but it has a certain impact to Thai society whatsoever. So I mentioned that its importance and then review a little bit what has been done on genetic improvement. And then in the last section, I merged into this paragraph. In this study, therefore, genetic parameters for body shape traits and deformity are reported for the first time. Okay, instead of saying that the purpose of this study is to study genetic parameter for body shape, that is plain. Since we have a kind of a very great achievement, because we will report this for the first time. So we mentioned that the information obtained can be used for planning a selection program to enhance growth of snakes in Burmese. The last sentence is very important. Furthermore, this study generates additional information that can enhance understanding of these traits because available information is currently limited. They not only applicable for the genetic improvement of snake skin Burmese, the information will be beneficial for the international arena by adding up the information for better understanding of this trait. So this look great, you know. One problem in writing introduction is that you always write back and forth. You go to the left and then you go back to the right and then you go to the left again. So writing this way is confusing and it fails in convincing the reader to go to reach the point, which is the objective of introduction. The readers will be distracted all the time. So to solve the problem, I suggest you to write down a key sentence of each paragraph and then elaborate. For example, like the previous example that I showed you, I might have written, I could not remember, I might have written the first sentence, importance of body shape trait. Okay, so, and then elaborate. So second paragraph is about the previous studies regarding this trait. Okay, and then you elaborate and keep the content within this framework. And when you move to another paragraph, never go back to the previous one. It doesn't mean that all information that you know should be put in this introduction. Not necessarily. Some information better take off. You put in, sometimes it devalue your writing. Okay, I have already mentioned this. Make the issue of global relevance by your writing style. Avoid general statement that doesn't have much meaning or already well known. Some people like to write in the I call it FAO style, like world population is forecast to reach 300,000 billion in the year, blah, 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 blah. So you spend almost 1/4 of the page, telling about something that is already known by people and it's not quite related to your issue. You can mention that it's important, but reduce to only one sentence or maybe 2 sentences. Some statistics, for example, annual production may trigger readers' interest. This is, I may use the wrong word, is not may trigger, is somewhat must, because many people always say like, this plant is very, is economically important. That words are quite vague, How important? My important and your important are different. So it's better to figure out, to put the data, the production data or the value, or if it's not high enough, but the trend is rising, then mention the rising trend or whatsoever, but always if I mean, you must provide something concrete evidence for that statement. Okay. Always have citations. Okay. Don't just write from your soul. This is scientific paper, not a novel. So every statement should have the origin, the citation, except things that are already known, like the sun rise on the east, went down in the west, something like that. Otherwise, you have to have citation. It should end with the objectives or the achievement of the study. Some manuscripts got rejected because they don't have objectives. These not frequently occur, but once in a while it happens. So don't forget, because to review your manuscript, one answer that we have to answer the journal is that, is the material method, the result comply or agree with the objectives. Okay, answer the objective or not. So if you don't have the objective there, this manuscript can be rejected right away. The other important issue is that in the review literature, you should cover works from authorized researchers in the field. What does it mean by authorized researchers? Means the famous one at that time, the one who published a lot on this issue. You can't miss their work. So as an editor-in-chief, what I always did when I went through a manuscript out of my field of expertise, I take the your keyword, and put in Google, and then so many publications would came out. And I check if in the introduction, have you mentioned these papers? Or if you just ignore all, of course, you cannot cover all in some case, and some publication may not have good quality. I would look at the one in the good journal and see, oh, wow, have you mentioned this? Did you miss this one? That is very important. And especially the authorized researcher, the famous one, is the potential reviewer for your manuscript, So if you ignore their works, it's going to be a problem. Introduction should not be too long. Some people, yesterday, I just suggest revising one manuscript because they have 4 pages introduction. That's too long. It must be clear and concise and convincing, okay? Reason 3 about the materials and methods. Actually, this part should not have too much problem, But if your manuscript use the obsolete techniques or approach, for example, like work that use the genetic marker. If you study genetic diversity and you still use the rapid, for ANRES, it depends. I will ask the opinion of section editor. But for big journals in Q1, they said clearly that they don't accept this technique anymore. Okay. Your manuscript with small experiment or small data set, it's not qualified. Or you already passed the first 2 bullets, but you have insufficient detail of materials and method. This is very important because if you don't give enough detail, then how can the reviewer understand your work and see whether your results is reliable or you have followed the correct scientific steps. So you need to write this in detail. Okay. Lack of improper statistical analysis. This is also happen very frequently. But I will leave this to Dr. Scott after break. Reason 4 is about results. Sometimes you get a great result, but you don't know how to explain it properly, It always happens with the complicated results. I have read in one book that they suggest that the good experiment is the experiment that try to design the experiment in a less complicated way. Some people put so many factors in hierarchy, pre-hierarchy, and then okay, you may use the appropriate statistical analysis method, but the way you explain the result and the way you discuss is a hell, very complicated. So better try to make it simple. Results sometimes were not appropriately explained especially when you explain the non-significant different result as if they are different. Okay, this is very serious. For example, people, this is about treatment 1, 2, 3, and then this is the ear weight of the form. The Mr. A may write down like T1 have highest ear weight, although it was not significant different with T2. I consider this wrong because they are not different. You can't say that this is highest, okay? So actually, you have to describe according to the result. T1 gave higher ear weight than T3, but it's not different with T2, and T2 and T3 are not different. You have to strictly explain according to the statistical result. The worst thing is that you interpret it wrongly and then you pick up the wrong interpretation into and go further for discussion. So that's misleading. And this happened in sometime in some international journal. I am not happy with this. Okay. If T1, T2, T3 were increment of dosage, you also have to mention the dose-dependent trend. Another point is that the data set that is better explained as graph should be presented as graph, then mention only important point in the text. Don't do like you are writing a thesis, some people just repeat everything, mention all the mean in the table. That is not the good writing, mention only the important and always show results of statistical tests. You have done performed the test already, so you have to show in either in both the table or in case you present as graph, you also have to show in the graph. Otherwise, how can the viewer check or review your result? Okay. For example, this is the, sorry, may I have a sip? Okay, so in this example, the author use herbs, eggs, mixing in the diet for the prawn, the shrimp. The concentration is different, is increasing from 0, which is the control, 0.5, 1, and 1.5 milligram per kg. Okay, they show the result with the main result is the SOD, which is the one parameter for immunity, and also show the weight gain in order to see that the purpose is to see the effect on immunity, but these things should not have a bad effect on weight gain. Okay. So the table is fine. Statistical test results will show. But look at this. If you make them into a graph, you see it clearly and see the trend better than in the table. Okay. So what I can see from this graph is that the use of these herbs increase the immunity in a dose-dependent manner. And when the dosage is excess, it has a bad effect. Okay, so if you look in the table, it's quite difficult to see. Look at this one. So I don't blame the author, because it's very difficult to show. They have a lot of cultivars, number of cultivars, and this box plot show the mean performance of each cultivar. So I asked the author, please put all the data in table. It's better. Okay. This is also, it's not readable. Actually, they perform the factorial design. So better show as table like this. It cannot be shown in a graph, too complicated. And there is some mistake in this also. Dr. Scott will mention about this later. Okay. All the means should be presented with standard deviation. Okay. People always mix up between standard deviation and standard error. We request you to put standard deviation because we want to know that how much variation is your data. If you show the standard error, it means that you get mean from each replication, 1, 2, 3, and then you calculate mean and standard error from this. So the value will be lower for standard error as compared to standard deviation. So we can't see the real fluctuation, the real variation of your raw data from standard error. Okay, there is an exception. Sometimes standard error can be used for the data on estimation, that based on estimation like heritability. Okay, reason 5, which is the last reason, and I think it's the most important, is that the discussion is poor, so that no new knowledge was generated. This is a pity. You have done a lot already. You collect data, you conduct experiment, you collect data, you did the analysis, you present the result, but then you could not discuss very well. So no new knowledge was generated. This is unacceptable. Okay. So discussion actually is not so difficult, if you know the concept. You have to discuss according to the objective or the hypothesis that you have before you conduct the experiment. Okay. Then you take all available information, which means empirical evidences and or theories to support or argue with your results, and then explain your results using the both information, empirical evidence, empirical data, or theory, and then draw conclusion. How do you want to convince the reader to believe in you? Okay, so by following these steps, discussion is not difficult anymore. Anyway, related issues that impact the reliability of the results must be discussed. You can't just ignore it. Okay, come back to the same, the previous example. In this result, I already mentioned that the result, there are three main findings from this data. Their hypothesis is that Earth's eggs enhance immunity. Okay, so in explaining the result, you should go right to the point that Earth's eggs can enhance immunity in a dose-dependent trend, but excessive dose cause adverse impact. Okay, so discuss according to the hypothesis. The result is yes, the herbs enhance immunity. Then explain why, why, how this herb enhance immunity. There are the studies that support or oppose your results. Okay, this herb has adverse effect at excess dose. You go the same thing. Is there any other report similar to yours or different to yours, and why? Okay, a dose-dependent response. Is this normal, or this is very abnormal? But there are still some other people that find similar thing with you, something like that. Okay, so it's like go by this pattern and then try to find, try to read as much as you can. The most important thing is all statements should have citations. All citation must be true. I used to reject one manuscript when I found that what they cite in the discussion was actually it's about another story, other thing. I don't have time to tell. That story is very funny. Avoid writing in this style. It's not incorrect, but it will make the reader or the viewer feel like, oh, this is not so professional. You know, Thai students love to write this way. Rustica et al. 2017 study female brood, blah blah blah, and similarly, Romachin et al. Study this. Is better to write up as your own style, and then use this citation to support that all evidence are really the published article. So better write like this value was comparable with the female brood collected from 3 locations subordinated bulk farming in West Java, and then cite all captured from the wild habitats, such as, you see, almost the same but different feeling. Okay, so try to avoid this style. For at the end of discussion, especially in the applied research, it is crucial to make recommendation according, and the recommendation should comply with what you have mentioned in the introduction. Okay, I have one example. These manuscripts say in the introduction that the information obtained would enable planning the efficient conservation measure. Okay, but in the recommendation, they say the information will be benefit the farmer to get higher income. It's different story, you know. So I asked them to revise. This is very important. Try to avoid meaningless comparisons. Some people just compare their result. Same as this guy, different with those guy. And then, so what? What do you want to point out? What do you want the reader to believe? Is, I call it the meaningless comparison, and avoid discussion on irrelevant issues. Some issue, something appear in your manuscript, but it's not the objective of your study. You haven't planned the experiment to answer that. So don't go down to have a very intensive discussion on that. That's unlikely. Okay. There is another issue that always happens. Some people repeat most of the result in the discussion. That is not okay. Okay, avoid repeating the results. So this is the example about irrelevant discussion. So this manuscript used the DNA technology to study something I can't remember. But in the discussion, they start by discussing the method of DNA extraction. In the experiment, they did not do compare the method or whatsoever, and this is not the objective of their study. You may mention that later, but don't emphasize that, you know. And if there are no issue on this DNA extraction, no need to mention it. Some people love to write discussion starting like a review, you know. I don't like this style. Some may be acceptable, but some will make me feel like, oh, this is the already know information. Like you start the same example, the Earth's X. This is I made up here. You may start discussion like, nowadays, herbs are widely used for health management of aquatic animals. Many species were reported to enhance immunity. Herbs has been used to enhance immunity of fish and blah, 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 blah. So I feel like, oh, this is the old issue. Okay, this is not new. There are so many literature on this, even though it's on the different fish, you know. So this is not a good way of writing discussion. This might devalue your manuscript regarding originality. Okay. Dr. Scott is already here. I will kind of spend 10 minutes more on this before break, okay? So conclusion is also very important. You draw out only major findings, and it must be exactly what you have presented in the result, okay? And then you make suggestions. Don't repeat all the findings. Draw out only major findings and then suggestion. Okay, I will go very quick on this. Writing abstract is also important, you know, because for some big journals, they don't have time. They look at the abstract only. If you don't have enough information on your manuscript, then your manuscript will get rejected. It is a short standalone summary of the work or paper that others can use as an overview. Okay. So you need to have mentioned one or two sentences on importance of the work and objectives. Okay, and then tell a little bit about materials and methods. In some kind of experiment, materials and methods is embedded in the result, that would be okay. You can go to the result right away. But most of the experiments, you need to clarify in brief the important materials and method, okay? And then you tell the result. Don't forget to include some important numerical data. And then most importantly, in your abstract with your main finding, okay? Reference, okay, very important. I will look at the reference. They must not be too old, At least 30% should be the recent one within 5 years back. Okay, this is my when I first joined Kasetsart University, it's my very first manuscript. So I got requested to revise the law. This is the way you respond to your reviewer, okay? Respond point by point. Don't skip any. If you agree, say you agree. You have changed from this to this. If you don't agree, explain why. No need to agree in every point, but you can't ignore a single point, that will make your manuscript rejected. That before sending out to the reviewers, we check the plagiarism of your manuscript. We will accept the plagiarism up to about 20%. 20% is a lot, about 20%. But we will look at the distribution of the plagiarized parts. If 20% came in two big parts, then you will get rejected. But if it dispersed, maybe you may be asked to revise. Don't co-submit. If I notify this, I will check you out. Co-submission means you submit your article, one single manuscript, to more than one journal. This is unethical. And at the moment, there is always a problem about preprints. People sometimes put the preprints in the website, and then when I check plagiarism, it would come out like 80%, 90%. It is ANRES policy that we don't accept this practice. The manuscript is not checked, is not correct, and then you post for citation is improper. This is our policy. So we would ask you to withdraw the preprint before submission, okay? Okay, so this is the end of my talk.
I will spend a short time for explain about what I have seen from the manuscript that's sent to ANRES. Mostly that lack of completeness of the explanation for the methodology and the result. Most of them are related to the basics or the information that the authors decide for the hypothesis test. Actually, most of the content in my slides related to what Dr. Uthairat just explained to you before. This is just what we have known so far, that we need to ask for this question before starting writing the manuscript. Most of the questions are related to every part of the manuscript. But what we found for the incomplete manuscript is that the explanation for methodology, it doesn't fit to the introduction or the hypothesis that the author would like to test. Sometimes we feel that because of the author would like to target results and discussion mostly more than methodology detail. But I would like to tell all of you that the methodology allow the audience or the reader to believe in the result that you report. So if you described the methodology well enough and acceptable, it will be help to support the results and the discussions a lot. Okay. So if we consider the structure of a research article, what I put the star on behind is what we found the incomplete information. Starting from the title, the title of some manuscripts doesn't explain or represent the details, or even it doesn't make any interest of the manuscript. Many titles will have information relating to the trade, the target populations, and also a new methodology that the author would like to present. Okay. And then the abstract, many abstract of the manuscript still have lack of enough important information to describe the methods or the methodology that used to test the hypothesis of the study. Actually, it should have enough information to tell the reader that what types of research that the author have been done for this manuscript. Suppose the author actually use some kind of experimental design. The author should also mention that their study was designed based on what, okay? And some of the manuscript or the study do not have any experiment, but they try to discover a new thing in the field. So the methodology to find those things and the new things is what they need to explain also. It doesn't mean that they need to explain in all detail, because in the methodology they also have spent up, enough area for describing the methodology. But in the abstract, they need to show the methodology that the author choose to use in this study. Okay. Then the introduction, some of the manuscripts that would like to compare the methodology, but they do not reveal any information related to those methodology that they choose to use, or they design a new one. I hope that you understand my presentation. Okay. It's likely if you want to compare the methodology, you need to review the methodology before and then come to the point that we need to find a new methodology that's more suitable or more efficient than the previous one. Even if you would like to test some treatment, the thing that you need to review is why you choose that level, that set of level of the treatment. Why not the other? Or even you use somebody else's methodology to test a new trade. So you need also mentions about the trade. Why you choose those trades to be studied with the methodology. And the most important thing in the methodology part or material method, most of the manuscripts still use some kind of explain about the software, which is not enough. The software is just a choice that you chose. Okay, it doesn't mean something that explain methodology that you use. So instead of using A software, B software, C program, or the other, you should mention about what methodology, what variable, what assumption for those variable, and how did you link those variables together in order to test the hypothesis? Suppose you use a kind of factorial design with block in the RCBD. So you need to describe what is the main effect, how many of them, how many subclass, how to consider the interaction between them, and also what is the block. Okay, and after that, you also need to explain the criteria that you use for considering the significant. Yes, for level or p-value. And if you want to compare the mean, you need to also describe about the tool that you compare mean, because they have many tools like t-test, Duncan, least significant difference, turkey test, student test. There are so many kinds, but you need to explain what you chose and, if possible, why. Okay. And then go to the result part. This part is not much information for the methodology, but it's related because if you describe for the methodology, suppose A, B, C, so you need to report the result from A, B, C. Consequently, by order, you cannot report C, A, and then B. Okay. So if you describe methodology in an order, you need to report the result in an order following what you describe in the methodology. Okay. And then conclusions. Okay. If the study is related to the statistics considerations, so you need to conclude in the right way of the statistics, which is, I think every one of you know already that you need to conclude based on the objective of the study. So this thing that I think everyone know, okay? But the point is here. When we talk about methodology explanations, okay, we normally have two things. The first thing is hypothesis, starting from the problem situation that you find out or you faced by your own, okay? And those hypotheses can be classified in many ways, but in many manuscripts that we have found is the hypothesis about relationship between the variable. Some of the manuscripts want to study factor affecting to the trade. I mean, mostly economic trade. And some of the hypothesis is about the differences either between trades, between variable, well, between locations, something like that. Okay. And the other that I did not put here is likely to recover or to discover something new. That is the other thing. Okay. Just explain, it might be enough in that way. But in this way, we faced with the probability. Okay, so we need to consider based on probability of the hypothesis. Okay, and if we talk about the research design before starting writing a manuscript, or even before writing, before starting the study, in the research design there are many, right? If we want to consider experiment, which most of you use that for finding new information. So in the experimental design, we normally consider experimental unit first, and then treatment, and then replications. If you talk about CRD and RCPD, they are different, just only experimental unit and how to classify them or manage them before assigning the treatment to them and then start the experiment. So this thing you need to describe some, okay? Because the different experiment, they require different information to explain by specific conditions. So if you understand the most important thing of the manuscript is used to be used worldwide, internationally, it means that thing that you did for the study should be repeat and get similar result, right? Otherwise, if it is very not, benefit enough. Okay. Secondly, is about the condition and limitations of your study. You need to explain if you do have. Okay. Why? Because this is the conditions and this is a, I would say, assumption or something that the reader should know before they consider the result. Because the result occur based on this condition and limitation. So we will understand the limitations and the conditions of the study, and they will understand and get the most use of the result that we report to them. Okay. The third thing is about types of data. We found many of the author that sometimes the data is frequency, but they try to describe in the other way. Okay. And sometimes the data is just binary. They assume the binary to be continuous and try the other way also. So each type of data require a specific study or specific considerations. If you want to link them together, suppose two variables that have different type, like a variable is binary and the other is continuous. For example, if we want to know their relationship, how could we estimate the relationship between them? Yes, we could estimate the relationship between a binary variable and continuous variable, but they have specific tools. So we need to describe the tool, not defend that it could be estimated or not. Because the leader just want to know how did you estimate the relationship between the two variables that have different type, right? So if you ignore that, the reader will have questions. How did you do that? Why don't you explain? Why don't you, this manuscript do not have information for that? And the other factor is the last thing, right? That like a covariance or adjusting factor of block. Block, this is the more important thing that we found many in the manuscript, because you use RCPD or block, but you do not, not you, many of the others did not explain about block. Okay, surely that in RCPD block is replication. We all know that, but what is the block? Okay, they should explain. Should be explained also. Okay, this I will spend a short time for explain. Okay, to explain them. Okay, you can ask the coordinator for the file. Okay, actually the methodology explanation that we use to describe the study, there are at least three groups. The first group is about experimental study, okay, which has the information that you need to answer like this. Okay, if you study, use some kind of experiment, control everything, just allow the treatment to be varied by the experimental unit. So you need to explain what is the experimental unit, how many of them, what is the treatment, how many of them, how to decide for that level. Okay, replications, how many of them? what is replications, block RCBD, interactions if factorial, and even time if you repeat measurement. We'll talk about this in the later slide. And then you should answer how to control those variations that are not the treatment. I mean, control the variant, okay, except for the treatment. How to randomly assign the treatment to the EU experimental unit, okay. And the other thing is how to test the hypothesis, how to test the effect of the treatment, how to compare the means, especially when the treatment has unbalanced experimental unit. I mean, not similar number of experimental unit, which you cannot compare them directly because they're not equal in terms of number of replication. So if you do that, you have unbiased comparison, right? Like that, okay? This talking about explanation only, okay? And here, the structure of experiment composed the set of treatment experimental unit, close the procedure and measurements. Okay, I will skip this, okay? I think you all know that. The second type of study is study in the populations, okay? The most important thing to describe starting in the methodology is the characteristic or structure of the populations, location, the size of the population, time to gather information from the population because it's dynamic, they change every time. So if you do not mention that, it means that you assume that the population doesn't change. It remains there until now, which is not true, right? So you need to mention about the time that you go, that you went to the locations and then gather information from the population. Okay? And the other is about general genetics or management or within the population. How many samples that you take from the population and you assure yourself that is enough for being a representative of the population. Okay. And then the same, how to test the hypothesis of your study, either associations, correlations, factor affecting the test. If you want to estimate something within the populations, how to estimate, how to test the hypothesis about that. Okay, predictions, trend analysis, and in the most manuscript mean comparison. And the last types of, that we skip, but there are many more than this, okay? It's field study. It's different from experimental study and also the population study. In field study, you cannot expect anything because in field, there are lots of variation from many factors, from many variables. So the thing that you need to start when you describe for this type of study is where. If you put GPS, geometric position system, you there, okay? Because it will indicate the height from level to sea, latitude and longitude, something like that. This is the information of the field area that you study and why you choose that field. Okay, and also the sample that could be representative. How did you gather the data from the field? Other related information that might be useful to consider when you test the hypothesis, because this is not experiment, this is a field. So we need to concern that all factors that being around the animal or plants will be vary all the times, okay? And the most important thing in the field study is statistics. Yeah, because this you cannot control anything. You need to use statistics that can describe good enough and allow you to focus on what you want to study from the field. Okay, so the model that represent things that related together and also the assumptions of the factor that might be related to the target trait or the target variable that you would like to study is important. So this requires a lot of explanations. I believe that you did good, but I doubt about the explanation. Okay, so you better explain enough for the leader, not for yourself. Okay. Here is, for example, something that many authors would like to know about two variables is the relationship or associations. But we do know that most of the parameter that we normally use to describe the relationship or association between the variable is correlation and also regression. But those two things is different. Are different. Correlations they calculated from covariance and a variance of a variable and the other. Okay. Actually, the length of the value of the correlation is between -1 to 1, and the regression is from infinity to infinity. So if you understand this, you will know that the two parameter is different. Correlations is about the relationship in a way of freely vary of one variable and the other variable. But ligations is likely linear associated between two variables. One variable changes one unit, and the other variable changes how much or how many, like that. So if you understand this, when you describe in terms of figure or in terms of value estimated from your data, it will be right. I mean, will be written correctly. Otherwise, some of manuscripts, some of the other, use the word that inappropriate. Okay? Sometimes they talk about correlation, sometimes they talk likely indication, but the two things are different. Okay. The other thing is about experimental design. Okay. The general thing is about CRD when the experimental units are similar. I mean, all of them. So you freely randomly assign the treatment to each one of them easily. But if the experimental unit can be classified because they are different, okay, you need to classify first and then assign the treatment to experimental unit within the block or within the group that you classify, and then process the study. Okay, so this is about the experimental unit between CRD and RCPD. But what we found from the manuscript is many authors do not describe about this. When they decide to use RCPD, they do not describe what are they different among the experimental unit. So can you expect the reader understand that? If you do not describe for the block, but you use RCPD, so can the reader understand what block that make you a problem? Instead of using CRD, you use RCPD. So like this. So my suggestion is just to describe for them. Okay, this is not a problem. This is what we do in statistics. If the experimental unit do not similar all of them, for all of them, we can classify them and then we set a block. Okay, but block is not variable. Okay, you do not need to explain, discuss, and focus on the block because block is a block. Okay, block is the arranging of the experimental unit in group. Okay, I describe for you here. You can find information from the website. This is normal. Okay, the most important thing is to describe. Okay, the other thing that we found a lot, okay, is confusions about factorial design. The factorial design we target on interaction effect between variable. Likely you consider two variable at least, and those variable has a different level or different subgroup. Suppose here the first line is about two variable A and B. You target on interaction between A and B. And then you have main effect A, main effect B. So you proceed the factorial design in CRD. Okay. If you have three variable, the second line. But if you, this is likely in plant side. Okay. If you have block, you put block inside, so it becomes two variable interaction or two variable factorial in RCBD. Okay. And again, if you have three variable, you have three variable interactions in RCPD. Okay. The graph here or figure here describe the difference that will be occur when you study using the factorial design. You see, what is the point of this slide is the factorial design normally target to interaction effect. So if you present that you use factorial design, mostly the target to divert interaction is significant or not. If the interaction between variable is significant, you have no need to mention about main effect. Why? Because the interactions is most important or more important than main effect. You cannot separate them because your analysis indicate that the interaction has significant enough. So main effect by itself seem not to be important. Okay. But if the interaction effect do not have any significant effect, then you can interpret main effect that has significant, something like that. So many manuscripts describe that they use factorial design, two variable, support 3/2 factorial design in RCBD. But in the result and discussion, they do not mention about interaction effect. They say about the p-value and then skip. They want to explain about main effect A or main effect B only. That is not appropriate. Okay. And the other thing that we found is about factorial design with one control, which is normally occur in plant science, plant research. Okay. The problem is not this can be used or not. Surely it can be used. Can be hypothesis tested. Sorry, because one researcher a report in horticulture science, okay, in 2003, that these types of incomplete factorial design or augment factorial can be used in a case of the author do not have enough money or have limitation, which they need to explain also in the material and method, why they choose to use augment factorial or incomplete factorial design. And also they need to describe the ANOVA that they use to test the hypothesis, either one-way ANOVA or two-way ANOVA. And also the comparison, which is complex. I would say that it's not easy. But if you have a colleague or co-author that have ability to test or to write a program that's flexible enough to handle this type of design, it's okay. But you need to explain. You cannot use complete factorial design way to explain incomplete factorial design. Okay, but many of the manuscripts just keep these parts and say that use 2/3 factorial with a control, and then that's it. That is not enough. Okay, this is rare case. Okay, please remember that. I would like to suggest you to use complete factorial design instead of incomplete factorial design. If you can find enough funding or you have resources for doing such study. Otherwise, you have many problems because this is like case and not many people know how to handle the unbiased comparisons and hypothesis tests. Okay, and even though you can handle it, the question is, can it be repeated for the result that you report? Okay, can we believe the result? Okay. The last set of the slide is about the some example when we buy from the manuscript. The first thing is CRD and factorial. If you consider the left-hand side here, experiment group, one control feed with 5 grams of A, 10 grams, 15 grams. Here, surely CRD, right? 1, 2, 3, 4 level from 0, 5, 10, and 15. Okay, so the effect is A, only A, treatment. Okay, A, 4 level, 0, 5, 10, 15 grams. Using a model for statistical test is y equal to mu plus A. Instead of A, we can put treatment, TRT, and then E. Okay, and when you describe, you just explain, you use CRD, consider A as a treatment at 4 level. Okay, and then mean comparison is about between subgroup of A's. This is simple and easy, right? Then experimental group control feed with no supplement. 5 grams of A but no B. 5 grams of B but no A. And then 5 grams of A and 5 grams of B. So it can be either CRD or factorial. But if you want to make the most benefit, you should consider factorial 2 by 2. So it's 2 level of A and 2 level of B. So you have A and B to be considered. No A, no B, 5 grams of A, 5 grams of B. See, you can consider this style like CRD, like above. But if you notice that they had a character likely allow you to consider them as a factorial, you can consider 2 by 2 here. And then the model change from this, the both y equal to mu plus A plus E to be y equal to mu plus A plus B plus AB plus E. This is a factorial design. The mean comparison is between A itself, level of A, between B itself, and then between interaction of A and B. You see, if you want to use CRD, the model will be this. Y equal to mu plus AB together. No A, no B, A, no B, B, no A, and A and B. So 4 level the same. But they has no main effect of A and no main effect of B. So just practice the same practice, but you couldn't make different assumptions and data analysis like this. Okay, so this is an example for your understanding that normally people confuse about practice and statistical analysis. Practice is practice, but statistical analysis is based on what you assume or make assumption for the practice. Both of them are support for hypothesis testing, but what detail of the hypothesis that you would like to test, that's the most important. Okay. The other case is about this. If you consider control with no supplements, but you use 5A, 5B, 10A, 5B, 5A, 10B, and 10A, 10B, it looks like all right, right? But if you force them to be factorial, it will be incomplete factorial automatically. Why? Because they miss off fit with 5A no B, 10A no B, 5B no A, and 10B no A. Actually, if you want to use factorial, there are two variables, right? A and B. But the level is 0, 5, and 10. So 2 by 3 by, 0, 5, 10, and A, B. So 2 by 3, right? Right? So there are some okay. It can be analyzed with the CRD here, and either factorial, but you need to know that incomplete factorial. But the other is completely different. If you set a control, no A, no B, but the level of B and A is different. So you have different level of A, but not equal to B. So it not really interact. Do you understand what I mean? The level is not really interact. Okay, it's specific. So if they are specific like this, you assume them to be specific. Even they look like combination, but it's specific. It means that you cannot ignore the specifications of those two. So you can assume the combination is one treatment, one group. So this could be just CRD. Okay, one group of control, one group of 3 grams of A, 7 grams of B. If you notice A and B, if you sum, they are equal to 10. Okay, 3, 7, 5, 5, 6, 4, 8, 2. They're not really, right? So you cannot assume that it's this main effect with free level, right? Like this. Okay, this is example. The other example is about times. This is occur either plant and animal. Okay, suppose you look at the picture. Suppose this is a corn, the maize corn. Okay, group 1 and group 2. You gather information from time 1 to time 8. Okay, and you know, people in general know that growth of each plant is different, are different, right? If you gather information like this, and then at the time 3, suppose you see time 3, they have some difference among the treatment. If you gather information from the time 4, can you assure yourself that the growth of time 4 will not receive any effect from the difference significantly different from time 3, from the previous measurement? Can you assure that? If you cannot assure, it means that those two groups, I mean group 1 and group 2, receive some effect from the previous measurement. It's not really, I mean, starting from the similar point like the beginning. So if you do not care about the times, it means that you bias. Okay. When you compare them biasly, what should be the result after that? Normally people ignore that, and then, okay, we know the bias of that, but we cannot know, we do not know how to handle it. So we put from time 1 to time 8, and then put the last time to compare, and the other will not report or do not discuss. That is I would say that it doesn't make any benefit to anyone, which the author should have. I mean, they should utilize the information benefit enough, okay, for themselves. So instead of ignoring measuring data between the times in the experiment, they consider times in the data analysis, like repeated measurement, either in CRD or RCPT. Okay, instead of allow just only one error or ratio, they understand that there might be some residual or error or residual effect that occur in between the times. And then they consider times in the effects of the trade that they measure. And after that, they can compare times, time 1 to time 8, and then compare treatment, a single treatment here, group 1 and group 2, and also to see the significance of times and treatment that might be occur. If they do not occur, it's good for you, but if they actually occur or do occur, then you explain for that. Okay? Because growth has different shape. So it might be the interaction between treatment and time. Like this, okay? Remember, I explained to you this did not tell you what you're wrong, but I just ask you to explain everything that you find or face in your study enough for the reader, okay? So in conclusion for statistical analysis, okay, mostly that, most problem that we found from the manuscript is how to describe data and variable. Author normally do not describe enough for variable. Okay. Hypothesis test, analysis of variance. Almost 90% of them do not describe for the model and assumption of data analysis. They describe just program and software. Okay. Mean comparison, they do not even why they choose Turkey, why they choose t-test, student test, why they choose least significant difference. But they need to explain a little bit, okay, for that. At least what type of tool that they use for mean comparison. It's just pair comparisons or multiple comparisons. Correlation, regression, estimation, prediction, and illustration, either graph, picture, or the other thing. And then the rest is tool. Okay. Please remember that we require just enough information, not too much information. Okay. And understandable for the descriptive explanation, acceptable and reliable for the methodology and hypothesis test, and then it should be cover and support the objective of the study. The other thing is related to Dr. Dailata explained to you this morning. I would like to mention some, okay, just to support. If you describe for the data in your study in the manuscript, normally the data described in terms of mean and standard deviation. Okay? Why? Because mean, when we describe mean, it likely we describe for the representative of the data set in our study of the group. Okay. And if we explain for the standard deviation, you know, right? Standard deviation powers square, power square is variance. Okay, variance is the square of standard deviation. So it represents variation in the group that you consider. Mean represents the group. Standard deviation tell you about similarity and variations within the group. So this will fulfill the reader to understand similarity among the group, within the group, among the group, and then the mean represent the group. Okay. Likely, if you estimate the value, you should put them with the standard error of its estimate. So you will know, the reader will know automatically that they can believe in the estimate value or not. If they have high standard error, the accuracy of the estimate value or reliability of the estimate value is lower than the less, the smaller standard error, right? The p-value also, you need to put the actual value that you got from the program or software you do not have necessarily to put either less than, equal to, or larger than. Why? Because if you put less than, equal to, larger than, you compare with alpha level, but you do not tell what you actually got from the data analysis for the p-value. So if you report the p-value, the reader understand well enough that if they would like to compare with 0.05, it will be less or more. If they would like to consider alpha 0.001, it will be less or more. So it allows the reader to make the most use from your report. If you put just only less than 0.05, the reader will have question, how much p-value actually? If they want more details about that, it doesn't support them to get that information, right? For the picture, for the figure here, it would be better if you indicate the significant between the group by putting superscript like in the table in the figure. But this is depend on your target for representing the information. Okay, you not have to, but it depends on the author, what the author would like to present from their data. Okay, but this is just a kind. I mean, this is a way to present things that easily to understand. They will know automatically different significantly or not. See? And I think the last two slides here, one or two slides here. Here, the left-hand side is about mean comparison. I would say multiple comparisons, similar to the right-hand side. Both those two tables use Duncan multiple range change. Okay, similar. But you notice, if you notice, here is the common style of manuscript that published everywhere in many journals here. Okay. But compared to the left-hand side, which is rarely found in the journal. But if you consider which one should be the most benefit for the reader, okay, I will go into the detail. Here they compare mean, multiple mean. They report just only mean, right? And then report the standard error of means. It means standard error of these 6 mean, or mean of the 6 treatment, and then report p-value less than 0.01. Yes, this is a common report. But if the author cares most about the reader, okay, they report mean, yes, but put the standard deviation inside. Okay, plus and minus. The reader will know automatically that, okay, this is uniform. This is not uniform. This is less variation. This has large variation. Okay, among the treatment and among the thread. The p-value they report exactly the p-value that they got from the computer or the software that they do analysis of the data. They know the detail. If they want to compare 0.5, 0.1, or 1.1, 10%, you know exactly that it's significant or not. But here it fixed, okay, less than 0.01, and how much? We do not know. Okay. So if I would like to suggest all of you to put detail that you could put in your manuscript, because the manuscript belongs to you. Okay, the benefit will go back to you. Why? Because the reader make the most use of your information. But compared to this, it just tell that it's significant or not. Nothing more. Okay. Similar, two groups with similar mean, it doesn't tell you that which one has uniform more than the other or better than the other. Okay. In industry, we would like to have uniformity, right? So here, you couldn't tell the reader. Okay, I think the last slide is here. So the question normally have been asked for reporting the result. If you talk about mean comparison, normally people ask, what are the value of the considered traits? Are they different? Is the difference different? I mean, significantly different. Sorry. So the target sentence that we would like to see is A larger than B, right? But we write a scientific article, so we should put information that indicate we did the data analysis in the right way. So we put A with mean and standard deviation like this, larger significantly or not than B is. So this is a complete information for the reader. Okay. Suppose that you want to open every piece of information, okay? But if you want to cover something, hide something, depend on the author, okay? And if we talk about the associations, normally do they have any associations? How much? How much accuracy is the associations significant? So suppose the sentence is A has relationship with B, but you need to put something from data analysis to support. The correlation equal to what? I mean the value. The correlation coefficients equal to what? And p-values. Can we believe in that value? Or the regression coefficient was the estimate value, how much the standard error of that estimated value and p-value. So it supports itself that we can believe or we can aware to consider that's right. Okay. And the last thing is we need to understand that things are always different. If you write a sentence, you need to understand that things are always different. Nothing's completely the same or similar. So when we talk about probability, the probability test will support the difference is significant or not. So the most thing is we need to declare the significance of the difference. Everything are different, but significantly or not, based on hypothesis or conditions of that study, okay? And the other thing is you need to understand if you want to translate your or discuss your result to the industry, sometimes different but not important. Sometimes even though they are not significantly different, but it creates a lot of importance. So this you need to show your understanding or your feeling of things that related to industry. Because you are the person who work intensively with your study, nobody know more than you. So you explain out from your experience from this study and discuss well enough to link to the business. Okay. The last thing is these three words create a lot of confusion and we found a lot of mistake of writing. Associations, relationship, correlation, and ligation. They are used to explain the link between two variables. Okay. But which one is support your target or which one is support your hypothesis? You need to choose the appropriate one. Okay, they're not the same. Please remember, they do not the same. Okay. That's it. I think based on the time that I prepare the information, I would like to stop presentation like this. I would like to answer and discuss with you.